Well, let's talk about pH. pH is an incredibly important concept in biology, and you can hardly escape it. Any living thing is orange. You know they're fairly acidic. If you eat yogurt, you know it has that tangy taste, and you can taste the acid. Vinegar is also very acidic. Probably one of the more acidic things in your kitchen. About the only base you'll find on the opposite end is baking soda in the kitchen. Most things we eat are acidic, and that seems to protect them from bacteria. So to be honest, I'm going to let the chemists really describe pH and why it's written funny. We'll take the simple route and just say that pH is a measure of the concentration of hydrogen ions. Right? So it's an ion of just hydrogen, basically a proton. Sometimes you'll see that if you put it in brackets, that means the concentration of it. And we'll leave the definition at that. So if we're going to measure something like pH, then we need a scale. Just like you measure things in centimeters, or feet, or grams, we need a scale and some units. And so pH scale goes from 0 to 14, 7 right in the middle. Seems kind of weird, that it's a bizarre scale. Well, it's about to get weirder. If we're to this side, the lower numbers, then we have an acid. And if we're higher than 7, then we have a base. Sometimes this is referred to as being alkaline. So either of those terms apply. We're used to acids, and we just talked about a few of them. The further this way we go, the stronger the acid. And the further this way we go, the stronger the base. We're used to acids being kind of bad, and we should be a little scared of them. But the same is true for a strong base. This would be like Drano or bleach. That can be pretty dangerous stuff. The happy place for us is right in the middle, right around 7. In fact, in the human body, our blood at least likes to be in a pretty narrow range between about 7.35 and 7.45. So that's a pretty narrow zone of tolerance. If we don't do that, then proteins start unraveling and changing, and the body stops working. All right, so just a little more about these numbers. Again, I'm going to let the chemist explain why this strange scale. It's just our job to memorize it. Here we have more hydrogen ions, and the concentration on the smaller the number, and here we have fewer hydrogen ions, and for some strange reason having to do with logarithmic scales, that number is larger. So already it's a little bit strange. If something gets acidic, we want to say that number goes up, but it doesn't. It goes down, it gets lower, closer to 0. And so the kind of right amount for us that our body is built, and most organisms are, is right around 7. That's just right in terms of the amount of hydrogen ions. And so a lot of bases, you'll see, have hydroxide ion, and so what that does, so if you add sodium hydroxide, the sodium is going to split off and contribute hydroxide ions. Those are going to absorb the hydrogen ions and make plain old water out of that. And so that means there's going to be fewer and fewer of these hydrogen ions in solution. Whereas an acid, on the other hand, something like hydrochloric acid, has a hydrogen and a chloride ion, stuck together by ionic bonds, but when you put them in water, they split apart. The chlorine ends up with the electron and that hydrogen ion splits off, contributing more of that to the solution and making it a stronger acid. But there's more. Not all acids and bases are the same. Common in a lab, you'd find hydrochloric acid, and every single one of these that you put in solution is going to give off a hydrogen ion. And so that's a strong acid. A weak acid is the familiar one that we just talked about, vinegar. And vinegar looks like this. Now there's a lot of covalent bonds there. This is an ionic bond and an electrolyte at that. So when you drop it into water, it forms ions. All right, what about vinegar here? Well, this is not an ionic bond. These hydrogens don't make any sense that they would give that up. They're pretty tightly bound to the carbon. If anything, this one is the most loosely held because some of those electrons are drawn back to these oxygens here. So this bond is kind of weak. What that means is if you drop one of these vinegar molecules into water, this hydrogen ion might come off or it might not. And so that makes it a weak acid. For every 100 of these you put in solution, only some of them are going to give up their hydrogen ion and create a more acidic solution, but plenty of them will still keep that hydrogen ion. So that makes it a weak acid. Now that we understand those, we can understand what a buffer is. And a buffer's job is to keep a stable pH. And so it does that either by absorbing or giving up hydrogen ions. And buffers can be set to different pHs depending on the mix of the chemicals. And so we might want to keep it at 7, but we might want a buffer that keeps our pH at 10. It really just depends on the circumstance and what chemicals we mix up. But a buffer is a combination of a weak acid and a weak base. And let me go back to that example that we used before of carbonic acid. So if this is carbonic acid, you might look at that and say, that's not that different from vinegar. Covalent bonds, yep, you're right. And these electrons are being pulled back toward the oxygen, so this bond is not all that stable. And so you'd expect that sometimes this gives up a hydrogen ion. Well, yeah, that fits the profile of a weak acid. So if we take that hydrogen off, then we have bicarbonate. I'm going to take it off of this one, and so we end up, if the hydrogen ion was given up, and so it would have been right in here, but we took it off, and so that electron is being held here. So this is an oxygen that is kind of in need of a friend, right? It gets 2 bonds like this one has. This oxygen's pretty happy, but this one's not exactly. And so this is a weak base. What does a weak base do? If you have hydrogen ions, bases absorb those hydrogen ions. And so a buffer, if the job is to keep a stable pH, and if we were to add a hydrogen ion to solution, it would want to bond to this weak base, the bicarbonate. And now that pH is neutralized. I didn't change my pH because I gave that hydrogen somewhere to go, so there's no more hydrogen ions. The concentration didn't change. What if I were to add a base, something with hydroxide ions, for example? If that's the case, that should absorb hydrogen ions and make it a stronger base. And in that case, the carbonic acid is going to give up this hydrogen ion so that this actually gets its way and forms water, but then this carbonic acid simply becomes the bicarbonate. And nothing actually changes in terms of the pH because my hydrogen ion concentration didn't change. So while this CO2 can lower the pH, if we have the bicarbonate, its conjugate base, you might hear that term sometimes, then it becomes a buffer, and these two can switch back and forth between these two forms and keep a pH stable. So a very important buffer system in the body along with phosphates and some others.